So my research team will be studying a condition called multiple sclerosis, or MS for short. So in this condition, the body's own immune cells starts to attack the nerves in the brain, the optic nerve or the eye nerves, as well as in the spinal cord. So with time, the nerve cells get injured and they function less well, and this can result in problems in walking, problems with bladder as well as bowel control, vision as well as cognition, which is the ability to think clearly. So when we look at MS brains that have been donated for research, what we find is that there's actually a significant amount of injury to the topmost layer of the brain. So this layer is also called the cortex, and what we find is that injury to the cortex is highly correlated with cognitive problems, problems with memory, as well as concentration, as well as processing speed. But the problem is that we are not sure why such injury in the cortex occurs, and there's currently no way to monitor this in living patients. So when we look at this brain model, we think of the brain as immediately lying just beneath the skull. But in actual fact, there is a covering that covers the brain, and this is actually called the meninges. The meninges actually protects the brain as well as nourishes the brain. But in multiple sclerosis, there is actually inflammation beneath the meninges. And it's believed that inflammation in the meninges is a key driver driving injury in the cortex. Well, when we look at nerve cells or neurons, and when we study them in school, it looked something like that, of course without the face. But in actual fact, if you look at this beautiful picture over here, it shows the complexity of what is going on within a neuron and around a neuron. And you can appreciate that it takes many components, many different parts for the neuron to function properly. So in order to understand how cells interact with one another, as well as the changes that occur within individual cells, we are applying this technique known as spatial transcriptomics. And this will allow us to understand what genes are being turned on and what genes are being turned off in the cortex and the meninges. And by understanding this, we'll be able to work out what is wrong, what is causing this injury in the cortex, and to let us find potential treatments for MS, and so that we can keep our neurons happy.